Hello students. Now this new topic will that the plant growth and the mineral nutrition. In this topic, most of the syllabus were omitted in this topic. Only the plant hormones remain there and for the 12th standard examination. So now here first you have to study about the growth hormones. So first this term hormone was coined first by Starling in the animal physiology. This term पहिल्यांदा coin केलेली आहे ती म्हणजे स्टार्लिंगनी. Now the plant hormones, this some factor which influence the plant growth. The factor which influence the plant growth, that is also called the hormone. That is called hormone, means the factor which is there and which influence the growth of the plant, so that is known as a hormone. Now the plant hormones inhibit, promote and modify the growth. Those plant hormones which are responsible for the inhibit, promote and modify the growth. Then here in this topic you have to study auxins, gibberellin, cytokinin, ethylene and abscisic acid. Out of these, auxins, gibberellins and cytokinin, which are growth promoters, which are called the growth promoters, and ethylene and abscisic acid, which are known as growth inhibitors and growth regulators. Growth inhibitors and growth regulators. But all the phytohormones which are called the growth regulators, all phytohormones are called the growth regulators. So now here what about the definition, exact definition of hormone? According to the Thiemann and the Pincus in 1948, the plant hormones are organic compound, naturally produced, naturally synthesized in the plant. Naturally synthesized in the plant. These are the plant hormones, means the organic compound, and naturally produced in the plant, which affect the growth, and other physiological functions at the site removed from its place of production and active in a very minute amount. And so this is about the definition of hormone. Hormone हे organic compound आहे, naturally synthesized in the plant and which affects the growth and other physiological functions at the site removed from its place to the production, and it is active in a minute amount. So this is about the definition of hormone. Then here you know that this auxin, it is the first hormone. It is first hormone to be discovered in the plant. It is the first hormone to be discovered in the plant and it is known as a auxin. The F.W. Wain used this term first auxin and isolated from urine of a person suffering from the pellagra. Pellagra, this is one of the diseases, urinary disease. And now here use this term first auxin isolated from urine of a person suffering from pellagra. Then natural auxin. Because the auxin, it is one of the plant hormone which is organic compound naturally produced in the plant, and natural auxin is indole 3 acetic acid. It is a natural auxin and it is responsible for the cell elongation and cell enlargement. It is the main important effect of this auxin. This is responsible for the cell elongation and enlargement. Synthetic hormones which are also produced are indole butyric acid, naphthalene acetic acid, and now the 2,4-dichlorophenolic acetic acid. There are 3 number of the hormones: indole and butyric acid, naphthalene acetic acid, 2,4-dichlorophenolic acetic acid, which are the synthetic hormones synthetic auxins. Then about this auxin. Auxin, it is to be the first hormone. It is to be discovered at the first time. And now here about this, many scientists also perform the experiment on the plant hormone. Now back to 19th century by the Charles Darwin. At the first time, he studied the tropism in a plant, and Canara, the coleoptile tip of the Canara grass. Now this is one of this experiment the performed by the Darwin on this canary grass. So now here this is a canary grass by the Charles Darwin. Coleoptile tip म्हणजे हा शब्द बरेचदा जाणार आहे. coleoptile tip. coleoptile tip means the stem apex जो आहे. coleoptile म्हणजे मोनोकॉटलिडेनसचा जो स्टेम अपेक्स आहे, which is covered by the sheath-like structure, so that is known as a coleoptile. So that tip, it is known as a coleoptile tip. Okay, the plumule tip, now it is nothing but the coleoptile tip. Now he exposed such a type of the tip towards the unilateral light, and he concluded that the something is present at the tip of the coleoptile which stimulates the growth, and that's why that this coleoptile tip bending towards the unilateral light. And now this was performed by the Charles Darwin, and he concluded that about that. Many scientists also performed the various experiments. Now about the Wendt experiment. So this Wendt experiments in 1948, approximately in 28. Now he performed the experiment, successfully isolated the auxin from the coleoptile tip of Avena sativa. Avena sativa म्हणजे वेंटनी जे मोनोकॉट प्लांट घेतलं ते म्हणजे Avena sativa हा ओट प्लांट आहे. ओट प्लांट, दिस इज अ scientific name of this plant, Avena sativa, and he selected this Avena sativa for this experiment and successfully isolated the auxin from the genus Avena sativa. So now he performed the experiment successfully also. Now this is the coleoptile stem apex of this. This is known as a coleoptile. This is a coleoptile tip, and this is the stump of this. And this coleoptile tip is cut off and which is placed on this agar block like this for some times, and then remove this coleoptile tip, and he take this agar block, and this agar block which is placed on the cut stump of coleoptile. This agar block, this is not the coleoptile tip. This is because a coleoptile tip which is put on the agar medium for some period. Then he removed this tip, and this agar block which is placed asymmetrically on the cut stump of Avena sativa like this, which is placed asymmetrically. Now it caused a bending. After some time it's caused a bending like this. So now here this is due to this, because he concluded that now in the coleoptile tip, now the growth hormone which is present there and which diffuses to the agar block, and when this agar block which is placed on this stump and which also showed the bending like this, so this much due to the presence of the auxin in that block, because which is diffused from the coleoptile tip, and in this coleoptile tip now the auxin which is present there. So this was also the experimentally proved that now the first auxin isolated from the Avena sativa by the Wendt. Then about this physiological effects and application of the auxin. There are so many physiological effects and application of auxin. So first of all, the apical dominance. Apical dominance, it stimulates the apical dominance. Apical dominance means though the apex of the stem. Suppose now this apex of the stem which continuously grow and which retard the growth of the lateral buds. It suppress the growth of the lateral buds. So that phenomenon which is known as the apical dominance. And the AUXIN promotes the apical dominance. Follow this. Apical dominance means these apical teeth or the apex of the stem continuously grow and suppress the growth of the lateral buds. Suppose now these lateral buds which are present here, this is one lateral bud and this is another lateral bud. But now only this stem apex continuously grow and stop or suppress the growth of the lateral bud. So this phenomenon, it is known as a apical dominance. It means that this is the first application of this AUXIN. It promotes the apical dominance. Then another second, 2,4-D means this is a synthetic AUXIN. Kills dicot weeds without affecting the monocots. Monocots crop plants जे आहे ते क्रॉप प्लांट्स मध्ये जर मध्ये जर डायकॉट प्लांट्स येत असेल आणि अननेसेसरी ते ग्रो होत असेल त्या ठिकाणी तर त्याला अशा त्याचं नाव दिस 2,4-D याचा स्प्रे मारला जातो आणि त्याच्यामुळे काय होतं की डायकॉट वीड्स किल होतात and without affecting the monocots crop plants. Seedless fruits are also the produced there. Parthenocarpic. Seedless fruits mean the carpinocarpic. Now this lemon, banana and this pineapple also. Such a type of the fruits which are also produced by the application of the AUXIN without formation of the seeds. So such a fruits which are also the produced there promotes the cell division in cambium. You know that this cambium, in the cambium now the meristematic cells which are present there and this AUXIN promotes the cell division in the cambium and which also responsible for the differentiation of the xylem and phloem and to the formation of the secondary growth in the plant. Now here this most important application of this AUXIN, it is responsible for the cell enlargement and the cell elongation. Then also the which AUXIN breaks the seed dormancy also and the bud dormancy. Due to this cell division, it promotes the cell division. It increases the rate of the respiration also during the germination. It increases the rate of the respiration during the germination also, which also happen due to the application of the AUXIN, which breaks the seed dormancy, which breaks the bud dormancy, which increase the rate of the seed germination also, increase the rate of the respiration also. What about this? This promote the cell division in this cambium. Now the spray of the synthetic AUXINs, which also promotes the flowering in the leech, flowering in the pineapple also. जर याचा स्प्रे सिंथेटिक ऑक्सिनचा जर स्प्रे तुम्ही स्प्रे केला असेल लीच प्लांट वरती किंवा पायनॅपल प्लांट वरती अर्ली फ्लावरिंग त्याच्यामध्ये तुम्हाला बघायला मिळतं अँड देर आर सो मेनी ॲप्लिकेशन्स बट दीज आर द मेन इम्पॉर्टंट ॲप्लिकेशन. So this is about the physiological effects and the application of AUXIN.